Hello, my name is Alex Bruin, and I would first like to thank everybody that made this symposium possible, as well as everyone that has made all of our research possible. Today, I will begin by talking about my research on using wastewater monitoring, or wastewater-based epidemiology, as a tool to track and look for genetic markers of disease, more specifically SARS-CoV-2 or COVID-19. Wastewater monitoring is an extremely valuable tool in disease tracking, and it is used around the world for various uses. My presentation will be focused on the wastewater-based epidemiology as a tool to track the community level of the spread of COVID-19. Now, this does not mean that COVID-19 is contractable through the wastewater. It just means that it can be traced through the wastewater. This is because the virus is not actually present and alive in the wastewater, but instead bits and pieces of its RNA fragments or gene copies are found in the wastewater. To prove the feasibility of this approach, which is tracking a disease through wastewater, a pilot project was launched this past school year studying the wastewater treatment plant in Star City, as well as sites around the WVU campus. The goal of this pilot project was to develop an early warning system for the community and university officials for COVID-19. This early warning system provides communities to better prepare themselves for the oncoming influx of disease and provides a near real-time analysis of COVID-19 concentrations in the community. Future aims of this project are focused on establishing a grander network of wastewater surveillance not only focused on COVID-19, but other diseases and their variants as well. So with the new variants of COVID emerging and the flu season that is approaching us, this remains a vital tool for our community. And because this approach is so versatile, we can also use it to track drug use in a community, which is another objective of this project. This can be focused on whatever drug that officials would like. For instance, we can use it to track the opioid epidemic in our state of West Virginia. Now moving on to the method sections, first up is the site selection. And this is where the places where the three different types of autosamplers are employed. If you can look at the bottom part of the screen, that is the ISCO 3700 autosampler, and this is an example of one of the sites that are selected. And sites are selected where there may be a larger population of infected individuals, or a larger concentration of the disease in question, or simply there are places where officials want more data or testing done on. Now going in deeper on the ISCO 3700 and the other autosamplers, they are suspended from the manhole so that they are out of harm's way of the wastewater that is flowing through the sewer. And in that sewer pipe, the autosampler takes what is known as a composite sample. And a composite sample is a sum of many different smaller samples over a period of time. In our case, those smaller samples were taken throughout one day, throughout the week. After wastewater samples are taken by the autosampler, they are then taken to the lab where they undergo analysis. Now, the genes in questions, the genes N1, N2, and RP of COVID-19, were found using an RT-ddPCR testing or assay. And then they were plotted in Excel to characterize and visualize the data. If you can reference this plot for the next part, after officials have received this data and it is plotted in the Excel plot right there, in this instance, they could see a spike in the plot where there is a continuing increase of concentrations of COVID-19, and they can then begin to decide what the next steps are. Are they going to put the community into a quarantine? More or less of a mask mandate? Are they going to lift social distancing directives, or will they employ more social distancing directives? Then the circle continues and they implement those guidelines on the community. Then we see how the community reacts to the guidelines. Are there less infections? Are there more infections? And then after that, the officials reassess and start the circle all over again. Now, this concludes my presentation, and I would just like to close up with one last statement that wastewater monitoring has emerged as and will continue to be a valuable tool for communities as it acts as an early warning system for the emergence and re-emergence of diseases and drug use. Now, thank you, and I look forward to answering any questions that you may have.